right, so that's Dr. Ben. Our next question is from Billy. Hello, again, Billy. Let's see. Can you ask Dr. Ben if there's been research on individuals on antivirals like HIV meds being less likely to contract COVID-19? Curious to know if something in antiviral meds or chemotherapy would affect this. Thank you. Yeah. All right. On the surface of it, the first reaction would be like, no, there's no way. They're totally different viruses virologically. But the answer is that some clinical trials have been done, and it's not actually a completely far-fetched idea. So the drugs that are being tested are two different kinds. So one set is a thing that looks a little bit like a nucleotide. So a nucleotide is going to be one of these bases of RNA or DNA. And the coronavirus tends to use RNA, and that's kind of the only thing that works. But these are all kind of misshapen versions, and sometimes you can get a DNA-like compound to work on an RNA virus, and vice versa. It has happened. Um, so for anti-HIV, they would tend to be DNA-like compounds. And for coronavirus, you would tend to want to go for RNA-like compounds. But the difference is one oxygen and one hydrogen on the compound in a particular place. And that difference is not always essential. So, yeah, it's not a terrible idea. The other class of drugs that have been looked at are these things called protease inhibitors, which would be part of the normal cocktail. So, if you had somebody who had HIV, they'd normally be taking 3 drugs at a time. It varies depending on the manufacturer, and there are some pills you can just take one little tiny pill once a day, and that's really nice. Usually, the formulation is 2 things that will hit an enzyme called the reverse transcriptase, which is the main self-copying enzyme of HIV, and one thing that hits the protease, and the protease is a protein-cutting enzyme. And the reason HIV has that is so that it can mature. Yeah. So HIV actually releases a little particle out of the cell, and once the particle has just come out, it's actually not ready to infect. It has to rearrange its insides quite dramatically. It builds this sort of thing that kind of looks like a sawed-off ice cream cone inside, and has 2 copies of its genome in it. And then it's got this multi-layered structure outside of that. It's really neat looking. Anyway, it starts out with all these things just locked together and arrayed just like that around the inner surface of the virus. And the protease comes along and snips them all in just the right spots so that they can be free to form up different shapes on the inside and make those different layers. And so, the protease inhibitors have been tested. And the HIV protease is really not that similar to the coronavirus protease. But remember, it's not just coronavirus enzymes that you can target. If you go after the enzymes that help coronavirus get into the cell, potentially that's a way to stop virus entry. And so these proteases would be things like TMPRSS2 and cathepsin L. That's just the name of 2 protein-cutting enzymes that are normally on different parts of your cell and can both help the coronavirus to enter your cell. And so the idea was that some of these protease inhibitors seem to have some degree of effect in stopping virus from getting into the cell, because they were working not just on the HIV enzyme, but also on the enzyme from the cell. Because to some extent, a protease is a protease, or there are these great big families of them, and there's only so many ways to build a working protease. And so, yeah, there's a little room for crosstalk between drugs for different things. Now, there was a big test. It came out very early. I think it was mostly done in China, and it was looking specifically at a combination of 2 anti-HIV drugs. I believe one was a protease inhibitor and the other was one of these nucleotide-like compounds. And anyway, they concluded that this was not an effective treatment. It didn't do anything. Didn't make anything worse the way hydroxychloroquine did, but it didn't make anything better either. And so it's like expensively doing nothing. Or really, it's like trying a thing to find out if it works. And then once you figure out, okay, it doesn't work very well, that's fine. You move on to the next thing because there are a lot more things that can be tried. And it's just a matter of lining up patients and doctors and hospitals to be able to try these things. That's the really slow part. So there we go. There's your answer. Thank you very much. This has been Ask Dr. Ben.